So I'm a plant biologist, so I'm going to talk about the application of CRISPR and related gene editing technologies on plants. So first, I want to explain why I'm learning this gene editing business, because some of the people in the audience know that I've been working on abiotic stress responses in plants for 20 years now, and we're still working on that because there's still many important unanswered questions. And more than 10 years ago, we got into epigenetics, and we're still working on that. But in the last few years, we kind of ventured into a little bit into gene editing, this technology development. So the reason is maybe can be explained here. In epigenetic studies, one of the pathways we look at is how small RNAs direct DNA methylation, how epigenetic modification happens at a specific sequence. That is, if you look at the genome, some sequences are methylated, some sequences are not. The question is why this particular sequence is methylated, the others are not. So we look at how methyl transfers are directed to a specific sequence, and also how demethylases are directed to specific sequences. And this turned out to be very complicated. So like this direction, so the enzyme is simple. DRM2, that's the DNA methyl transferase that carries out de novo DNA methylation. But in order for this enzyme to function at a particular locus, you would need many dozens of proteins to achieve this. So this includes the production of non-coding RNAs, in long non-coding RNAs, and small interfering RNAs. And then these small interfering RNAs, of course, goes into argonaut as effectors. But these argonaut, they don't really directly cause DNA methylation. Somehow, the argonaut would have to recruit this DNA methyl transferase eventually, but this is still not understood. But there are many proteins involved that's known that involves this chromatin remodeling complex, this DDR complex, and also involves this POR5 complex that produces a scaffold RNA. So on and so forth, and also the POR2 is also involved in some of the process, and another different complex. So it's so complicated. At the beginning, we were looking at the possibility of reconstituting this pathway in a test tube, but now after many years, we are still looking at it as as a fantasy. But so clearly, you can see from its previous two talks, it's not as simple as a small RNA guiding a DNA nucleus directly to the target DNA. So it's more complicated. So for this reason, we wanted to be able to find simple ways to target DNA methyl transferases and or other enzymes in epigenetic regulation to specific sequences, so we can do sequence-specific modifications, epigenetic modifications. So that's the original purpose. And so let us to this targeted gene editing. So let me some of the background I guess it's not necessary anymore because of the two excellent talks before this talk. So if you look at a lot of us rely on genetic variation for our studies. And, you have natural variation or induced chemical or otherwise induced mutagenesis. But these all have their advantages and disadvantages. So a lot of us would want to have a way to make a targeted DNA modifications or to create variations in the sequence we are interested in. So this is already explained by Patrick. So one can take advantage of the cell DNA repair machinery. So when you create a double-strand break, and things will happen either to make mutations or to make gene corrections, depending on homology combination or not. So to target DNA nucleases to specific sequences, there are different ways. A number of years ago, people have been trying with zinc fingers, and then about 3-4 years ago, people started to look at the tail proteins, and then, of course, now the attention pretty much shifted to the CRISPR system. In all these three systems that people have been using, you have this DNA binding module that zinc finger DNA binding protein, the tail protein, or the non-coding RNA as a guide to guide nucleases, fork1 or Cas9. So zinc finger has been used, but not by so many labs. It's a pretty tricky to engineer zinc fingers to bind to your sequence of interest. So I understand. I'm not sure. I don't think there are more than a few dozen labs who have been able to do this. So, but yeah. So that's why when this easier protein came up, tail protein, people get really excited. So the tail proteins are also known as tail effectors, transcription activator-like effectors. These were identified originally from Xanthomonas genus of bacterial pathogens of plants. These bacteria using the type 3 injection system that was discovered by Xinyang here at MSU that injects these effectors, many different proteins as effectors into the plant host cells. One type of the effectors are the tail effectors. They can act as transcription activators to activate the expression of genes that will benefit the pathogens. And the tail protein has a really interesting structure in the sense that the DNA binding module has these repeats. Each repeat has about 34, 35 nucleic amino acids long, and they repeat pretty conservatively, except in the middle two amino acids. It's called di amino acid variant that specifies the DNA binding specificity. So in 2000, so when it's known that 2009 that this code was solved, so it was found that one repeat would recognize one nucleotide, and then there is this really nice one-to-one code between these dinucleotides and the nucleotide sequence of the target gene that it binds to. So, of course, now we know the structural basis of this specific DNA recognition by tail proteins. Is we collaborated on this work with Yan Ning's lab at Tsinghua University. It's a really beautiful structure that shows how these tail proteins bind to specific DNA sequence. So we also found that this binding to the DNA sequence is influenced by DNA methylation. So there is a different code for recognition of C that's different from MC. So MC is recognized pretty much like a T. So this one would have taken this into consideration when you design tail proteins to bind to your target sequence, because in many genes in plants, and of course in mammals, and in some of the microbes, they are methylated. So that would influence the design of your tail protein. So so originally my intention was to use the tail protein to direct DNA metatransferases, DNA demethylases, and histone modification enzymes for sequence-specific epigenetic modifications. But we still been doing that. We're not publishing anything yet because it's a long-term project. But in the meantime, we also tried to use the tail protein to target other effectors, other functional domains like fork1 DNA nuclease from bacteria for this gene editing, and we showed in 2000, well, published in early 2012, that you can do this, target a protein, a gene, in this case, a transgene, a Gas transgene that is preceded by this stop codon, so it's non-functional. But if you make a tail to target here to create double-strand break, then through DNA repair and this stop codon can be removed, then you can achieve expression of this Gas reporter gene. Of course, now we've been, being in Yang's lab at Iowa State University, have published the papers in showing that you can use the tail nucleus to edit genes in rice. And we've been trying to do that also in rice. And then this, you can look at this, these two genes that we targeted, for example, MST7 and MST8. You can get deletions, most of the time deletions. But you can see here that the efficiency is not very high. It's 10 out of 151 T0 transgenic plants, we got mutations in the target gene, and this 4 out of 75. This, because it's pretty good. It's not very high, as I will show you later. That with CRISPR, it's much higher than this. But already, this is a lot of improvement compared to zinc finger nucleus. Because zinc finger nucleus in plants, I think, it's been applied for more than 20 years, and still only a few genes being targeted, and only a few events being reported. So this, but tail nucleus is a lot easier than that. And we've also published papers to showing that you can design schemes to facilitate this design of engineer the tail protein to bind to your sequence of interest. And also we reported that we can fuse a transcriptional repressor, a very short 12 amino acid peptide known to function as very nicely as a repressor of gene expression to the end of this tail protein, and then this can function as to target to any gene to repress gene expression. For in this case, we target to this reporter gene promoter, and we see in all the lines, very nice repression of this expression of this gene. So most people know that the tail proteins are from Xanthomonas bacteria, but actually there are other genus of bacteria that also have tail protein or tail-like proteins. We also worked on this Rastonia tail-like proteins, and I think some interesting properties that may help, may be complementary to the tail from Xanthomonas. So clearly, tail could be used to direct nucleus and to also, for gene repression or gene activation. Of course, already tail protein itself is the transcriptional activator, and we've been trying to fuse tail protein to other functional domains. So there are a lot of things you can do with tail protein. That's why I think 2012 this is tail nucleus was considered as one of the top 10 breakthroughs by Science magazine. But I can see that very quickly, next year, then was replaced by CRISPR-Cas. So things really happen very fast. I don't have to go through these slides anymore. I guess so. Yeah, we can skip this. We can skip this. So you already know what happened, the history. So I just want to point out that the first papers showing the application of CRISPR-Cas in human cells was in January last year. Then very quickly, a few months later, papers of showing application in other organisms, including plants, showed up. Including 2 from our group. In fact, just 1 month after this publication, as soon as we received we got this Cas9 plasmid from Zhang. Within 2-3 weeks, we were able to see that in plant cells, plant chloroplasts, it works really nicely. It works really well. But of course, just working in the chloroplasts is really not good enough. But let me first explain what we did in plants. In plants, we have to use basically just have to change the promoters. So of course, we use the plant U6 promoter. This is the Arabidopsis thaliana U6 promoter to drive the expression of the single guide RNA. And we use the double certified S virus promoter, constitutive promoter to drive the expression of Cas9. And in rice, we use the same certified S promoter for Cas9 expression, but use the rice U6 promoter. But in our group, there's an independent study from another student who wanted to try the different promoters, different U6 promoters, and also different promoter to drive the expression of Cas9 using the ubiquitin promoter. It works actually just as well, or sometimes even better than the certified S promoter. So both are available to anyone who requested. We've sent out to at least more than 100 labs around the world for their applications. So as I said, within 2-3 weeks, we were able to show that in chloroplasts, this is really the cell system in plants. You can see that it functioned really well when we targeted this YFFP gene to remove the central parts to create a YFP gene through homological recombination. You just need to make a double-stranded break here by targeting a single guide RNA to this region. So we were able to show that this happens at quite a good frequency, almost 5% compared to YFP transformation alone, So, but of course, the real test is in plants, in transgenic plants. So we designed single guide RNAs to target many different genes. This shows 2 examples here. 3 against BRI1, GAI, and GS. These are genes that are very important for plant hormonal responses, so they give very obvious phenotypes, so easy to see the effects. So we designed the single guide RNA, of course, taking into consideration that you need the PAM motif, NGG. And we found already in the chloroplasts, the PAM is important, but it's not essential actually. In the chloroplast, at least, you can disrupt this NGG. The efficiency is reduced, a lot, but it's not, it still works. So in some instances where you cannot find an NGG in your sequence, then you may have to live with a low efficiency, but it still can work. So I think that's good to know. Then we also often, try to design a target where we can find a restriction site at the target site, so we can to be easier to detect the sequence changes induced by the CRISPR-Cas, as I'll show you later. So this is one example. So because there is a restriction site within the target sequence, so you can see that without editing, you can see a cut of the genomic sequence. And with the editing, these are different T1 transgenic lines. You can see, they contain the wild type sequence that are cut, but also they contain a sequence that's not cut, that indicating this sequence has been edited. So these are a few numbers here. If you look at this, just T1, this is the first generation after you collect plants, transgenic Arabidopsis that express Cas9. You can see that the frequency is pretty high. Out of 19 T1 transgenic plants, you can have 16 plants that you can detect convincingly there are mutations, by sequencing, by whatever means. So this is here, 49 out of 60. So it's a very good frequency compared to tail that I showed you earlier on in the slides. And of course, meantime, other groups also tried this in plants, and I think pretty much the efficiency is in this range. And of course, we also tried play different things, like including targeting, 2 sites to see if we can delete the fragment. Indeed, we saw, deletion of fragment of a few hundred base pairs, close to 1 kb. We have not tried to delete longer fragments. But also we saw inversions, so all kinds of changes. So I think so all can be useful depending on your purpose. So like us, several other groups reported at the same time. You can really, express Cas9 and the guide RNA in plants, and it works. You can get T1 plants that have mutations you can find. But I think I have not seen, everybody just stops there pretty much, but we know that T1 plants, it's not enough. We don't know, these mutations are heritable or not. What's going to happen in the next generation? So we asked that question, and we found actually, first of all, I have to tell you that in T1 plants, we checked, many hundreds, probably more than 1,000 transgenic T1 plants targeting different genes, and none of them has any homozygous mutations. All of them are chimeric. So that means they have mutations in, 2 or more mutations. So actually, they are. We have plants showing that they are mosaic. They are not just chimeric. They are mosaic. So when you go to many of the mutations, most of the mutations, when you go to the next generation, they disappear. They're not there anymore. And some of them are still there. And the new mutations occur in the T2 generation. So in T2 generation, of course, you also have a chimeric. I didn't show you here, but then you can start to see the useful ones. The useful ones are those the homozygous ones or biallelic ones or heterozygous. They can be heritable. So these are the useful ones. In T2, you start to see them. So T1, you cannot see them. You see mutations, but you don't know whether they're going to be useful or not. So my recommendation is, although everybody is eager, when you try this on your gene, you are eager to see mutation in T1. You're going to really easy to see mutations, but it's useless. So you can waste a lot of time. So just go directly to T2 and look for homozygous or biallelic or heterozygous plants. So this will save you a lot of effort. And then if you go to the next generation, T3, then you really know for sure they are homozygous or biallelic or heterozygous. No, I wanted to show you. So even in sometimes, like this one, we targeted I guess this is a BRY1 gene, this receptor for the plant hormone, brasinolide plant hormone. When you target this gene, and they cannot perceive brasinolide, and the plants grow really small, so they have dwarf plants. And in T1, you see some plants showing dwarf phenotype. You are so excited, you think I have the phenotype. You think it could be homozygous or biallelic. It's not. It turned out none of these plants are homozygous or biallelic, even though they show the phenotype. Because the reason is that they don't have the most mutations are not in the germ cells, but in the somatic cells. The majority of somatic cells are mutated, so they cannot perceive brasinolide. Still see the phenotype. So the phenotype can be deceiving. So I just want to point this out, because a lot of times the people look at the T1 plant, see plants with phenotype, and say, "Wow, okay, this is the plant." Actually, you can be disappointed in the next generation, and the phenotype will disappear, and then some real phenotype is going to appear, then it'll be heritable ones. That's what you want, So So I also want to show you this example to tell you that, in the target sequence, you can see, most of the time insertion and deletions. Deletions can be a few base pairs, can be a little long. And the insertions are mostly 1 or 2 base pairs. In rice, we only see these insertions of 1 nucleotide insertions. So, so in the T2 next generation, you can really see the phenotypes, the dwarf phenotypes. Then these are real ones. These are heritable ones. So you can check there's only 1 mutation, or if they're biallelic, only 2 mutations. There are no other mutations. So the question of specificity, of course, is very important. It's a question on everybody's mind. And we also asked this question, but we did not do any systematic experiment to test, how the variation in the guide RNA going to influence the specificity. But from the, number of guide RNAs we've designed, clearly, consistent with what Patrick showed, that the seed sequence is very important. And so far, I only find one case that outside the seed sequence, you can see one nucleotide mismatch. You can still get the targeting to work, but we really tried hard to look for off targets, and for this we initially tried to most of the time we tried to do whole genome whole genome resequencing. So we sequence the genome, then of course when you resequence any plant, even the wild type plant, you never done anything to the plants. You're gonna once you say sequence, you analyze, you're gonna find you compare to the sequence in the database, you're gonna see lots of thousands of indels and SNPs because sequence errors and also natural mutations and variations. So the question is how to distinguish these from real off targets. So we did this by 2 ways. By one way, just really to look at we figured that the off targets are most likely going to have some homology to the guide RNA. So either we use the guide RNA 20 base pair guide RNA sequence, or use the 12 base pair the seed sequence to get a list of putative off targets. And then we, at the putative off target, we really check, you eyeball checking to see if any of them have mutations in the whole genome resequencing. So this shows you this, the sequence coverage. So I think it's pretty deep, so about 60x coverage. And you can see that if you look at the target region, the sequence of interest, this is the guide RNA target sequence. You can see clearly most of the sequence. This is the T1. You can see T1 is not homozygous. So you can most of the time you can see deletions of different lengths or insertions. So most in a majority of the sequence. So it's not difficult to find the mutations in your target sequence by resequencing. It's very, very obvious. Of course, this only shows the partial sequence. I can't list the 60 sequences here, So, but it's another target sequence. You can see, changes here, the target sequence. Another example. But the thing is that we look at the putative off targets. The sequences that have very limited homology to 20 base pair guide RNA or the 12 base pair seed sequence. None of them we could find any mutations, at least from the 6x coverage. So if we sequence deeper, 1,000 cover X, we may see some rare mutations. It's possible, but we have not done that. So so far we can conclude, I guess, there is no widespread off targets in plants in Arabidopsis. In rice also, we sequence many rice transgenic plants, about 50, 60x coverage. We don't see just off targets. We don't see them. Doesn't mean they are not there. They're just not very frequent, or they're not widespread. So why this case we can only speculate. I can just tell you that there might be some differences between plants and animals in this regard because, for example, microRNAs, it's well known, 1 microRNA in animals can target 100, 200, or even several hundred mRNAs. That's well known. But in plants, 1 microRNA can only target 1 or a few target RNA that have perfect or near perfect homology to the microRNA. So microRNA is much more specific than in plants than in animals. Why is the case? Probably has to do with the Argonaut, but may have other factors that are also important. In this case, Cas9 is the same Cas9, but so we really don't know why the difference. But yeah, it appears to be more specific in plants. So this showing an example in rice. You can see this is a T0 first generation rice transgenic plants. You can see phenotypes. You can detect the mutations. Design is similar. You need a PAM. You want to try to get restriction site so you can detect the mutations more easily. For example, this is one example. This is the wild type Cas equal type rice. You can have an enzyme cut here. With engineering, with the editing, then you see the plants without this enzyme cut that's been edited. You can see in here clearly there's this one plant. Does not seem to have any wild type sequence left. So in your sequence, you can see actually indeed it's a homozygous plant. So there's no wild type sequences. Every sequence is mutated. And you go to the next generation, this is heritable. And there's just no other mutations. So rice is different. Rice in the first generation, T0, first generation, you can get homozygous or biallelic plants or heterozygous plants that are useful plants. And why this rice transformation is different from Arabidopsis. They have to do when the germ cells has expressed Cas9 and when the target sequence in the germ cells get edited. So this showing one example here. This is in rice, as mentioned already. This is the biallelic plants. You have insertion, and in this one copy you have insertion A. In another copy you have insertion T. So insertion so far we find only one base pair in rice. In Arabidopsis it can be different, but mostly are one base pair. But there are also other insertions. But this in rice only one base pair. Yeah, you can also have deletions, So you can see phenotypes, you can see mutations, and some of them can be heritable. So this is the important difference between rice and Arabidopsis. So in rice, you get a chimeric or mosaic ones, but also biallelic and homozygous. And these homozygous biallelic can be inherited to the T1 generation. So they're useful plants. So this shows you one example. So this is a heterozygous plant. You can see it's segregated in the next generation in the T1 plant. This is the homozygous. From T0 is already homozygous. You can see, in the next generation, it's completely, it's still homozygous, still this phenotype. So we also did, this genome resequencing of these rice plants. As I mentioned already, we don't find off targets by this method. So we actually, this is quite useful. One of the genes we targeted, when the gene is edited, you have this albino leaves. So using this phenotype, we were able to reconstruct, the pattern of embryogenesis in rice, depending on how early the cells get edited. So you can look at, if they are edited at the first embryogenic cell, and then you can get, the homozygous plant, albino plants without any green segments. And if they get, edited later on, you can get, mosaics of different, showing different patterns. So I think it can be useful for developmental studies, these kind of things. Yeah. So. Of course, one of the most attractive feature of CRISPR is this multiplex possibility of multiplexing, And this is especially important for plants because the plants, compared to other genomes, are known to have many gene duplication events in evolutionary history. And many plant genes we study are in gene families of several or even dozens of genes. And it's a headache for everyone to study these genes because they have redundancy, this issue of redundancy. So to make high order mutants, it takes a lot of effort. So now we have a good way to target multiple genes using one construct. We tried to hook up different guide RNAs on one construct. So far we have a construct that express 6 guide RNAs on one construct. And each, of course, guide RNA may target one gene, or if you design it in a way that the right sequence, you can target multiple genes. So it has the potential to target many genes. And we also, still this is not finished. We are trying to facilitate the process by putting this into a gateway system. But the real excitement, of course, it's already been shown by the previous speakers to be able to not just make mutations, to really to precisely change the sequence. So we've been trying to do that, and we can do that as well easily as in animal cells, as these guys showed, in protoplasts in the plant cell system. But that's not useful for any of us. I mean, you can, so for protoplasts, you can introduce guide RNA, introduce Cas9, then introduce your donor DNA that with whatever sequence you want to be inserted or to be replaced with. But the problem with plants is, whole plants is how do you deliver the donor DNA? You can't. You can't just, plants don't have an egg you can inject, this mRNA for, the donor DNA or, mRNA for Cas9 or, So that's the difficult part. And so people are trying different ways to introduce the donor DNA. The easiest is to introduce in a typical a binary vector, but you only have one copy inserted there, so it's not, its efficiency very low. So to increase the efficiency, you could use well, also the fact is in plants the vast majority non-homologous end joining repair. So homologous recombination is very rare. So to increase the efficiency, one would have to do positive negative selection to select the rare events of homologous recombination. And also, if this combines with the CRISPR-Cas, to increase, the efficiency by creating double-strand breaks, I think that would help a lot. And also, we are trying different ways to deliver donor DNA by using viral sequences, by other means, I don't want to talk about here yet. So I think this is a real excitement, but this is really the bottleneck in plants. I think once this problem is solved, we can efficiently deliver donor DNA to, you want to add a tag to a protein, so you have this protein is expressed and it's a native promoter, you can, or change it, the promoter or the, enhancer, as Daniel showed, or change a particular amino acid to make the, enzyme more efficient or whatever. So I think that will be doable once this problem is resolved, and then that's the day we all hope, to see. So. Getting back to my original purpose, we are still interested in using these modules as TAIL or CRISPR and Cas to direct different functional proteins for gene regulation for genome modifications. And Patrick already showed you that you can disable the Cas9 so that you can make DNA cuts, and then you can fuse different proteins to this dead Cas9 to activate genes, to repress genes, methylated genes or demethylated genes. So this is what we've been playing, but this is a long-term project, as I said, still, but really exciting, a lot of potential. Okay, I'll stop here and acknowledge people who did the work. So the gene editing work started when I took a leave at this new university in Saudi Arabia. That's like 4 years ago now. And I stopped 2 years ago, so these people in my lab, this now, he's an independent PI there now, so he's continuing the project. And then this CRISPR project was started last year in my new lab in Shanghai, so I'm building a new institute in Shanghai within the Chinese Academy of Sciences. It's known as the Shanghai Center for Plant Stress Biology. The Cas9 plasmid was originally provided by Hong Zhang's lab, and we collaborated with Ning Yan's lab on the study of tail structure and how it's recognized methylated DNA. Thank you for your attention.